It's my pleasure to welcome you to the 2011-2012 Sigma Xi Local Honors Lecture. Sigma Xi is an international research society for research scientists and engineers. It was started in 1886, has about 500 chapters worldwide. The chapter at Iowa State was the 34th chapter, and it was the first land-grant chapter started in the Sigma Xi organization. Membership comes by way of nomination through one of the Sigma Xi members. So if you're interested, please contact me or one of the Sigma Xi members. You can find more information about Sigma Xi on the Iowa State website. Well, it's my pleasure to introduce the speaker for this evening, Professor Surya Malapragada. She is chair of the Department of Chemical and Biological Engineering. She's also the Stanley Chair in Interdisciplinary Engineering here at Iowa State University. She has a number of honors and made wonderful progress in her career development. Her research focuses on the development of new polymeric materials for medical applications, including neural tissue engineering, controlled drug and gene delivery, and as templates for biomineralization. The work has implications for cancer therapy, to nerve regeneration, to single-dose vaccines. Dr. Malapragada is a fellow of the American Association for the Advancement of Science, American Institute of Medical and Biological Engineering. She is a recipient of the NSF Early Career Award and was named one of the world's top 100 young innovators in 2002 by MIT's Technology Review magazine. She received her PhD in chemical engineering from Purdue University. So please join me in welcoming Dr. Malapragada to the lecture.
I'd like to thank Dr. Minion for the very nice introduction, and also thank you for the invitation today. It's really my pleasure to be here, and it's a much larger crowd than I was expecting, to be honest, so on this cold wintry evening. So glad to have you all here, and hopefully you'll find something interesting in the lecture. I know it's a very diverse audience. There's some folks here from a speech class, I understand, so it'll be interesting to convey the information to a really broad audience. So what I'd like to do today is to give you a little bit of introduction to what we do in our group. We're really focused on polymers. And again, since it's a pretty broad audience, most of you are familiar with plastics, of course. Polymers are just plastics. When you think of plastics, you think about cheap commodity materials. These are a little more designer materials, as we like to think of them, designed for with particular applications in mind. And what we are looking at is ones that have a micro or nanostructure that could be tailored for specific applications. We do it in a variety of different ways. We use patterning techniques like micro and nano patterning, or use other types of morphological control to control this micro or nanostructure. Also take advantage of chemistry to do that, so use appropriate synthetic routes in order to get the materials with these desired properties. And we're looking at two different broad areas. One of them is in biomedical applications. So once we make these materials for specific biomedical applications, we're also looking at the inverse, so to speak. So how can we learn from nature and make materials similar to what we see in nature, which could be used for biological or other types of applications as well. And in all of these cases, we are interested in seeing how the molecular properties affect the macroscopic behavior and vice versa. I'd like you to just interrupt me at any time if you have any questions. You don't have to wait till the end to ask questions. One of the areas that I won't talk about much in this talk today that we've done a lot of is in the area of neural tissue engineering, but it gives you a good illustration of what I was just talking about, so I just wanted to spend a few minutes on this. So in this case, what we've done is take these polymers. In this case, these are degradable materials because we don't want something that will stay in the body forever and ever. So these are materials that can fall apart after, say, depending upon the chemistry, a few months or a few weeks, and that's something that we can control. So we have these pattern materials that we're trying to use for nerve regeneration. So in this case, the nerves that we're talking about are peripheral nerves, so those are the nerves that go to your arms and feet, not the spinal cord we're talking about. So if you have a transection in the nerve, if you have some damage to the nerve because of injury or illness and so on, if you think about a gap in the nerve, then the cells on one side cannot grow towards the other side and bridge the gap because they're not getting the cues from the other side, if this gap is, say, more than a centimeter or so. So really the function of these materials that we are designing is to be able to guide them in the right direction. So that is shown in this figure here. If these cells are just grown on a regular substrate, they grow in all sorts of directions. But if they are on these micropattern substrates, you can see that they're very aligned and growing in a particular direction. That's what we want here, right? Because we want them to be able to bridge this gap. And so the idea is pretty simple. We have this material that's made into a conduit or a cylinder with these micropatterns on the inside so as to guide the growth of these cells and be able to essentially bridge the gap. And this is a study that was done in rats, and we can see that this is the cross-section of this regenerated nerve. So initially it's empty space, and at the end of 6 or 8 weeks, we have this regenerated nerve facilitated by these materials, essentially. And there's also functional recovery. The rats can walk now. They cannot once the nerve is transected. So it gives you an illustration of what's possible with these materials. Right now we're also doing a lot of work with adult stem cells and trying to see if we can incorporate these in these conduits and so on. But I'm not going to talk much about this project, but it just gives you an illustration of what I was talking about. What I'm going to focus on today is to talk about some materials that we've developed and two different applications. One is in gene delivery and the other one is in biomineralization. And these materials are a little different from the degradable materials that I just showed you with the nerve regeneration project. In this case, we are developing some materials that can respond to surrounding environmental cues and do some things which are important for these applications. Okay, you don't need to focus on the actual structure itself, but I wanted to put it up there for those of you who have a background. But what we are making are essentially what are called block copolymers. So what that means is that you have different repeating units within the molecule that we are making. So you have 100 of these repeating units, 100 of these repeating units, and so on, all linked together to form this long chain. So this polymer molecule is this really long chain. In this case, it has a molecular weight of 12,000. And this is just a single molecule. And we design it with these different building blocks so that we can get the kinds of responses that we want from this material. What is it that we're looking for? We're looking for something that can dissolve in water. And so this is the free polymer or free polymer in solution, aqueous solutions. And as you increase the temperature, what it does is, because of some parts that like water, some parts that don't like water, so the blue parts don't like water. So when this molecule is in water, the blue parts want to stay away from the rest of water. So they form what's called a micelle, which is a nanometer scale feature, which then can assemble further as you increase temperature and concentration to form an actual physical gel. Now, in addition, this material has these other green blocks which confer upon it the ability to respond to changes in the acidity of the environment. So if you change the pH and make it more acidic, less acidic, you can have differences in the properties of this material. So this can dissolve faster or slower depending upon the acidity of the surrounding environment. So this is a molecule that we've synthesized which has both the ability to sense the temperature around it and respond to form these gels, or sense the acidity around it and respond accordingly. And so this is a diagram which shows you as you change the concentration and temperature here, it goes from, and we've just added a blue dye so you can see it clearly. This is just a liquid solution, and now it turns into this gel. As you raise the temperature, as you at this particular concentration, so you dissolve the polymer, 28% of the polymer in water. As you go from, say, 10 degrees centigrade to around 37 degrees centigrade, which is physiological temperature, so your body temperature is 37 degrees centigrade, it forms a gel. Why is this important? Why is this interesting? So we want to be able to use these as for a couple of different applications. One of them is as templates for mineralization. I'll talk about this after I talk about these two. So if you have a material that is liquid at room temperature or below body temperatures, and then you inject it into your body subcutaneously, intramuscularly, whatever, forms a gel. So it forms a solid. So you can this solid will then dissolve slowly over time and release whatever is trapped inside. And so you have a drug delivery depot, essentially. So if you have some drugs that you want to release over a long period of time, you can use this as a fairly simple way to inject a depot inside your body, which will then release slowly. You don't have to have a surgery to put this depot in. You don't have to have a surgery to take it out because it's just a simple injection. We're also looking at it for gene therapy. So the idea is, instead of just delivering small molecular weight drug molecules or protein molecules, which you can do using this method, can you also deliver genetic material which can then be expressed by cells and used for gene therapy? So that's another motivation for doing this. A third one is, can we use this polymer to also grow some inorganic materials, nanocrystals around it, and form a nanocomposite? And so now we're not talking about just an injectable polymer, but also an injectable nanocomposite. Polymers themselves might not have the mechanical properties that you sometimes want. So if you're looking for some load-bearing tissue, like cartilage or bone and so on, it's not just organic material. You also have inorganic nanocrystals like hydroxyapatite calcium phosphate nanocrystals in it. So can you use these as templates to grow those inorganic nanocrystals and have injectable nanocomposites? So that's sort of the idea behind this. So that's what I'm going to focus on. First, I'll talk about the use of these materials, or looking at these materials for gene delivery, and then talk about biomineralization. So what is gene therapy? The idea is to deliver therapeutic genes instead of delivering protein drugs or small molecular weight drugs. Right now, I mean, this is a little older. It's from 2007, but it hasn't changed that much in terms of the composition of the different clinical trials that are ongoing in gene therapy right now. Most of them focus on cancer at this point. So that's been a huge target of the gene therapy area. And of course, there are several other applications as well. But there's a lot of interest in being able to try to treat cancer using gene therapy approaches. Now, most of the work with gene therapy has involved viruses, because viruses do such a good job of delivering genetic material. Right? That's what they do. But there are lots of drawbacks associated with viruses, as you can imagine. One is immunogenicity, potential immune response that that your body can have based on the administration of the viruses. So it's always difficult to deliver a second dose, and There have been some high-profile deaths linked to some gene therapy clinical trials involving viruses, so it's come under a lot of scrutiny. So our approach has been to look at polymers as delivery devices for gene therapy instead of viruses. Of course, they're not as efficient as viral vectors, but they don't have some of the drawbacks associated with it, so it's in some ways a safer alternative. And we, as well as others, are looking at different polymers that can do the job as far as gene delivery is concerned. These are some of the different polymers, common polymers that have been investigated a lot for gene delivery. Again, the issue here is that polymers are not as efficient, not very efficient at delivering genetic material, so how can we increase the efficiency of transfection? So in all of these cases, these are what are called cationic polymers. Usually, cationic polymers are the ones that are explored for gene delivery because you have this negatively charged DNA. These cationic polymers can be positively charged usually, and so they form a complex which is called a polyplex. So that's the general idea. So you form this polyplex which has this condensed DNA which you can then deliver into cells. Since a lot of work is centered around cancer therapies, of course, one of the things that we'd like to do is to deliver this therapy directly to cancer cells, preferentially over other types of cells. And that's the biggest issue with chemotherapy. You know, if you don't have the targeting capability, you do a lot of damage to normal cells in addition to the cancer cells that you're trying to target. So the idea is, can we do things like deliver suicide genes to cancer cells specifically without affecting the normal cells around it? And there have been a lot of approaches that have been used for this targeting. Most of them rely on looking at the surface of the cells, and so there are some differences. For instance, there's some overexpression of, say, EGF, which is epidermal growth factor receptors on the surface of cancer cells. So people have tried to exploit that to look at targeting cancer cells specifically as opposed to normal cells and so on. What we are looking at is being able to try to exploit differences within the cellular environment, not just on the cell surface, to see if we can use that. For instance, if you look at this compartment within the cell, which is called the lysosome, there's a difference in the acidity or the pH level of within cancer cells versus more non-cancer cells. And so our thought was to try to exploit that, because that's a significant barrier for gene delivery. So if you look at a cartoon of just a cell, and this is the polyplex, which is the polymer DNA complex we are talking about, it needs to get into the cell, and then the cell has what's called an endolysosome, which is a compartment with a low pH environment where most of the DNA the DNA that's being delivered here can get degraded. So what the polymer needs to do is to protect that DNA in this step so that it can then get back into the cytoplasm and then into the nucleus where it could be expressed. And once it's expressed, then you have the products that you're looking for. So there are three main barriers intracellularly that you can think of. One is actually getting into the cell. The second one is endosomal escape, getting out of the endosome without much degradation of the DNA, and then getting into the nucleus where it can then be expressed. So this is the major challenge. So can we try to overcome these barriers and develop this polymeric gene delivery system that can selectively target cancer cells, and also have this polymer that can serve not just as a gene delivery vector, but also as a sustained release mechanism. So that's where the gelation properties come in. So you can think about looking at tumors that are maybe accessible, so you can inject directly into tumors. And so you can have the intertumoral injections where you have this depot that's formed. And now you have sustained delivery. As the polymer is dissolving, or as this gel is dissolving, you can release these polymer DNA complexes over time, because with the polymers you get only transient expression anyway. It's not a permanent change to your genome or anything like that. So if you have this continuous supply of these polyplexes, then you can produce or express those genes. So we've actually made a family of different, the polymer structure that I showed, the cationic groups, different cationic groups to have control over the chemistry. And why these particular pentablock copolymers? What advantages do they have? One of them is the ability to form those micelles that I talked about. Now, these micelles are formed because you have some amphiphilic, it's an amphiphilic molecule which has some hydrophobic and hydrophilic parts, water-loving and water-hating parts. And if you look at our cell membrane, it's also amphiphilic. So this micellar structure is really good in being able to integrate with the cell membrane and facilitate cellular entry. And there have been studies that have shown that micellar structures like these can help promote entry into the cells. So that's one of the barriers that we talked about. We have this thermoreversible gelation property, which allows for this sustained release over time. Again, compared to viruses, you don't have as many of the safety and efficacy issues, potentially. And again, the cationic character allows it to form these complexes with DNA because of the positive and negative charges of the two molecules. And then we also have these we synthesize these polymers, and we can also have reactive terminal ends which can attach to specific biomolecules if you still wanted to do some of the conventional targeting approaches as well, like EGF and so on. Okay, so we made these polyplexes, which is pretty simple. You just mix the polymer and DNA together, and you end up forming these polyplexes. I don't know if you can see it from the back of the room or not, but this is a cryotransmission electron microscopy image of these polyplexes. The scale bar here is about 50 nanometers. You see these structures that are formed. This is an atomic force microscopy image. Again, this scale bar here is about 100 nanometers, so you can see that they form these pretty condensed structures. And we've done a lot of comparisons with XGen, which is also a polymeric delivery vector, but it's very toxic. Some of the cationic polymers have a lot of toxicity associated with them. So even though XGen is very efficient in gene transfection, it has a lot of toxicity associated with it. With these pentablock copolymers, we have minimal toxicity, as you can see here. And if you look at the size of these molecules, we can have these fairly stable size ranges even in the presence of serum proteins, because that's another issue. If you have these charged molecules lying around in serum proteins, then you have a lot of potential aggregation and things like that can happen. And then these 100 nanometer structures actually don't stay as nanostructures, but they become larger aggregates, which can never get into cells. But here we can see that even in serum proteins, we can maintain the stability of size stability. So the polymer itself forms gels, but can these polymer DNA complexes form gels instead? And we've shown that yes, they do. And then they can dissolve slowly, releasing these polyplexes as a function of time. And the question is, what are we releasing? As this gel is dissolving, are we getting free polymer, free DNA, or are we getting these polymer DNA complexes? So we've done some agarose gel electrophoresis, which shows that whatever is released from these gels is still in a complex state. So if it's free DNA, then it moves this way. If it's complexed, you can see that the bands stay right here. And so we are releasing polymer DNA complexes as this gel is dissolving, which is exactly what we want. And so we've done some in vitro transfection studies, so taking cells in a petri dish and looking at how well, using a reporter gene, in this case, a gene that codes for green fluorescent protein, we can see that we have transfection that's pretty comparable to XGen in this case, which is our standard that we're using, even in the presence of serum proteins. And We've done some in vivo studies. These are subcutaneous tumors on mice, and we can do intratumoral injections where we can localize this gene expression right here using a luciferase gene. And we also wanted to look at being able to selectively transfect cancer cells even if it's not localized delivery. So can we get the selectivity of transfection that we are looking for, specifically in cancer cells versus non-cancer cells? To do that, we looked at this co-culture setup where we have a cell line, which is a cancer cell line, which is stably transduced to express, to show up as red. And then if we have a co-culture of normal cells and cancer cells, then we can look at the difference between transfection that's occurring in cancer cells versus in the non-cancer cell lines. And in all of these cases, whether we do co-cultures or separate cultures, you can see that we get selectivity of transfection specifically in the cancer cell line as opposed to the normal cell line, which is what we were expecting. But if you look at our control samples, for instance, using XGen, you don't get the selectivity of transfection. It's almost equally good whether you're looking at cancer cells versus non-cancer cells. So in order to look at the mechanism behind the selectivity of transfection, this is a really busy slide, so don't worry about the details here. But what we're trying to do is the top two graphs correspond to cancer cell lines, and the bottom two correspond to non-cancer cell lines. And what we have here is the transfection that you see initially, and those are the colored bars and these hatched bars. And as you add chloroquine, which is an agent that's known to help endosomal escape, as you remember the different intracellular barriers, one of them is this vesicle inside the cell, which has a low pH environment, which degrades the DNA, and one of the functions of the polymer is to allow it to escape from this. We see that there's an increase in the transfection, and chloroquine is an agent that's known to help endosomal escape. So when you add chloroquine, you see an increase in transfection efficiency. But for the non-cancer cell lines, you see this huge increase in transfection efficiency as you add chloroquine. So you can see that in these non-cancer cell lines, the endosomal escape is much more of an issue, and addition of chloroquine bumps it up much more compared to what you see in cancer cells. So you see the increase in gene expression is almost 200-fold higher in normal cells than in cancer cells if you add chloroquine. So what this means is that the differences that we are seeing in transfection between cancer cells and normal cells is largely because of this difference in endosomal escape. And why is that? Because there's a difference in the pH of the environment. For the non-cancer cells, it's a much lower pH. For the cancer cells, your endosomal pH, lysosomal pH is much higher, and so there's lesser damage. And so what we have here is a polymeric delivery vector that allows for selectivity of transfection based upon the differences in the intracellular pH environment of cancer cells versus non-cancer cells. And so what we can do is get the sustained release, get the selectivity of transfection using this approach. And lastly, I also want to say we can also co-deliver not the gene as well as some small molecular weight drugs like paclitaxel, which is a widely used anti-cancer drug. It's fairly hydrophobic. What we have is a system that forms micelles, and so that's great for delivery of hydrophobic drugs. Micelles are actually, a good way to understand it is soap. Soap forms micelles, for instance. That's how you wash away the dirt. You form these micellar structures where you can trap the dirt and that's washed off. So you have this hydrophobic environment within the micelles, which is great for loading hydrophobic drugs for delivery. And there have been studies that have shown, and the mechanism is not fully understood, that if you have a co-delivery of gene and drug, there's some synergistic effect that goes on where you can actually increase your gene transfection efficiency because of the presence of the drug. So we can use the exact same delivery vector that we are using, not just for the gene, but also for delivering these anti-cancer drugs. A double punch, essentially. And we surely see the synergistic effect. If you have this paclitaxel loading, it doesn't seem to matter how much is loaded, whether it's 20 or 50 weight percent. You see this increase in the transfection efficiency because of the presence of the paclitaxel. And so this way we can deliver not just the genetic material, but also the anti-cancer drugs using the same delivery device. So just to summarize this part of the talk, hopefully I've shown you that we've developed some injectable sustained gene delivery systems that can either be used for localized delivery to tumors if the tumors are accessible, but even if they're not, they have the selectivity of transfection, so they can be injected intravenously at lower concentrations, but still can maintain selectivity of transfection for cancer cells versus non-cancer cells. They have good transfection efficiency, but less toxicity compared to other polymeric vectors, but we are still not at the level of viral vectors, definitely. Again, this provides a paradigm for designing targeting vectors based on looking at intracellular differences, like pH differences, as opposed to just cell surface markers that have been used for targeting cancer cells versus non-cancer cells. I want to switch gears a little bit and talk about the same family of polymers, but in the context of biomineralization. So what exactly am I referring to? There are several examples of biomineralization in nature. A good example is bone, where you have collagen fibers which template the growth of inorganic nanocrystals like hydroxyapatite, which is a form of calcium phosphate. Again, there are several other examples with calcium carbonates and so on, mollusk shells, things like that, which, again, the organic molecules template the growth of the inorganic phase. This is another beautiful example. This is from Joanna Eisenberg's work. This is a glass sea sponge skeleton, which has this intricate structure. This is a centimeter. You can see different length scales here. This is 1 centimeter, and this is 5 millimeters. You see this intricate structure, which leads to its wonderful strength to weight ratio. And having this hierarchical assembly at multiple length scales is so important for getting these kinds of properties. Of course, that's the holy grail. We are not at this level of control yet in terms of the self-assembly process. But again, what we are trying to do here is to take these polymers in aqueous solutions, which form these micelles, but then attach mineralization proteins or peptides to the ends of the chains that control the formation of this inorganic phase. The idea is that then you can have these mineralization proteins or peptides control the formation of this phase, while the polymer template actually controls the placement inside this elastic matrix. So if you have these micelles, you can selectively coat them with the inorganic phase and then have them self-assemble further to form these solid structures. So in addition to forming the gels that I showed you, they actually form these elastic solids. This is completely by self-assembly. Just by changing the pH and the temperature of the solution, you can get them to form these nice cylinders. It's a completely reversible process. And if you look at the structure of these solids that are formed using a variety of X-ray scattering and other techniques, you see very ordered structures. This is indicative of what's called a hexagonal closed pack structure, which is formed because of these micelles packing together in a particular arrangement. So this in turn has a huge effect on its thermal stability, mechanical properties. So if you just take the bulk polymer itself, this is how it behaves. If you take the self-assembled solid, you can see that it has, this is mechanical testing. This is what's called a storage modulus. And even at very higher temperatures, this one goes only to 40 degrees. This is at 80 degrees. You still see high storage modulus. And so you can see that it has very different properties just because of the self-assembly process. In addition to the cationic ones that I showed before, we've also looked at some anionic, which are negatively charged, essentially, or zwitterionic, which have positive and negative charges. And using these as templates, they also have the temperature sensitivity, similar to what I showed earlier, but then you have different functional groups here, which can be used for different types of the mineralization conditions. So what we have here is an injectable nanocomposite. So it's not just an injectable polymer anymore, it's an injectable polymer with inorganic nanocrystals. And this is just an image of these inorganic nanocrystals, in this case calcium phosphate or hydroxyapatite nanocrystals. This is about 50 nanometers, the scale bar, and you form these long elongated nanocrystals, and they are all packed between these polymer micelles. So if you look at the structure and packing of these micelles to form the solid, you don't disrupt that because of the formation of the inorganic phase. It's all in between the micelles, which is exactly where it should be. Otherwise, it's not really a nanocomposite. You just have a precipitate of the inorganic phase, and you have the organic phase separately, which is not what we're looking for. This is a true nanocomposite. So we've made a variety of these different nanocomposites with different kinds of ordering based on the different templates that we've used, the anionic, the zwitterionic, and so on. Why is this important again? This is actually a project that we are doing with some folks at the University of Iowa in their orthopedic department, a medical school. They came to us and said they wanted a material. They're looking at patients who have undergone trauma. So if you have damage to the cartilage, if you've been in an accident and you've had severe damage to your cartilage, you have cracks in the cartilage, they want something that they can just inject into the joint, stabilize the joint, something that can fill the cracks in the cartilage and have mechanical properties similar to cartilage, so that you don't develop post-traumatic osteoarthritis. They have this huge center on post-traumatic osteoarthritis. So that was the request they came to us with, which is not an easy one to fulfill, because cartilage has very unique properties. It's very difficult to match the properties of cartilage. So with just organic materials alone, it's hard to do that. So we came up with this idea of this injectable nanocomposite, and we've shown that since it's injectable, since it's liquid at room temperature, and then when you inject it into the body, it immediately forms this gel nanocomposite. What that allows us to do is to be able to fill the cracks. So you can have different shaped cracks. If you start off with a material that's already solid, you can't fill cracks. But this way you can actually fill the cracks. And we've shown that the properties, both compressive properties as well as shear properties, are very similar to that of native cartilage with these nanocomposite materials. And it also forms a good bond with the cartilage, because you have these nanocrystals that are growing in situ in the body, essentially, and so they can sort of bridge the gap, and some of them grow into the cartilage itself and provide good bonding, because otherwise it's very difficult, again, to find materials that can form good bonds with cartilage. So this is an ongoing project where we are, again, they're doing further tests in whole joint models to look at these injectable materials that we use for prevent post-traumatic osteoarthritis. Yeah.
What's the longevity of the injection? Can you vary it? And I assume the bone eventually replaces what you've injected, right?
Yeah, that's a good question. This is not meant to be a permanent replacement. They've done some studies to show that, of course, when you have trauma and impact, there are some cells, chondrocytes out of the cartilage cells. The ones that are directly impacted, they die. But there are these cells in the surrounding areas, which can be rescued if mechanically and biochemically stabilized immediately. And so that's what we're targeting. And so they are looking for a material that will stay in the body maybe for 15, 20 days to provide that kind of stabilization and rescue those chondrocytes so they don't die. And so that's sort of the general idea. Another area where we're looking at, so in this case we're looking at inspiration from nature, bone particularly, to develop these injectable nanocomposite materials. Another area of inspiration for us are these magnetotactic bacteria, which are these bacteria which have these chains of magnetic nanocrystals in them. This is a close-up of those. And so we've worked with some proteins that are in these magnetosome membranes surrounding these particles, and using that in conjunction with our polymers. And that protein, one of the proteins is MMS6. That's the one we've worked with. We've shown that we can make these magnetic nanocrystals outside of the bacteria. They form these beautiful, nice, uniform magnetic nanocrystals. While magnetite is found in several living species, we've shown that we can go beyond what's found in living organisms. We can make, say, cobalt ferrite crystals, which are not really found in living organisms. But again, this is completely room temperature synthesis. You can form these nice platelet-like crystals, completely in aqueous solutions. These nanocrystals have a different magnetic properties compared to the magnetite crystals. And so we have a lot of versatility in where we can go with this bio-inspired synthesis route, making more complex magnetic materials using these mineralization proteins from these bacteria. Again, if you look at the magnetic properties of the crystals that are formed using this MMS6 protein, you can see there's conjugated to the polymers. You can see there's a huge difference. They have the highest blocking temperatures, highest magnetic irreversibility at high temperatures, which are all indicative of well-formed nanocrystals. Very interesting magnetic properties. Again, you can think about these magnetic nanocrystals in this elastic polymer matrix for artificial muscles and all sorts of different applications that way. So in summary, for this part of the work, hopefully I've shown you that we can learn from nature, use proteins from nature or peptides based on using approaches similar to nature to use polymer templates attached to biomolecules to form these nanocomposites, whether it's the calcium phosphate nanocomposites or magnetic nanocomposites and so on. Again, this is completely room temperature synthesis, no heating or post-synthesis annealing required to get these highly crystalline samples, which is very unusual. Typically you have to heat, but if you heat, then you destroy the polymer. So this is the only approach to form these elastic polymer nanocomposites with this method. And we can control not just the crystal shape and size, but also the placement of the nanocrystals in the elastic matrix, which is a nice thing to do to control properties. So finally, I'd like to acknowledge all of the wonderful students and postdocs who worked with me on all the work that I showed. They're the ones who actually did the work, and so they deserve the full credit. But I also would like to thank all of the collaborators, numerous collaborators that I've had and have, without whom this work would not be possible, both at Iowa State and elsewhere. And of course, funding from different agencies who pay for all this work, again, to make it possible. And thank you for your attention. I'll be happy to answer any further questions.